Hello everyone. Information Box Ticket Lifestyle brings you food microbiology, food poisoning caused by Giardia species, Foodborne Giardiasis. Before continuing to this video, don't forget to like this video, subscribe to my channel and press the bell icon button so you don't miss any of my videos. So in this topic we will cover sources of contamination of foodborne giardiasis, characteristics of giardia, clinical manifestations of foodborne giardiasis, pathogenesis of giardia species, epidemiology of foodborne giardiasis, detection methods of giardia species, prevention and control measures. First, we will start with its introduction. Diseases connected to diarrheal illnesses are brought on by parasites which are widely dispersed in the environment. The sickness is mostly linked to parasites that are water and food-borne, and these parasites represent the biggest threat to food production. The greatest threat to human health and economic loss to the country is posed by parasitic protozoa, particularly in underdeveloped countries. Cryptosporidium, Cyclospora, Giardia, and Toxoplasma are a few of the parasitic protozoans that pose a concern to humans. The main parasites known to cause human disease is Giardia, and each year several epidemic cases are documented. Giardia duodenalis, Giardia lamblia, Giardia intestinalis, and Giardia muris are the giardiasis causing species that affect people and other mammals. Although there are many cases of foodborne parasites each year, there aren't very many cases recorded, which may be because surveillance and detection methods aren't particularly good. Sources of foodborne giardiasis contamination. The primary source of contamination is the excrements of domesticated animals, wild animals and cattle, and it is often spread by fecal-oral pathway. Giardia cysts may also be found in shallow spring water and sewage effluent. These cysts are very contagious and can survive for a long time in cold, moist environment. Giardia may withstand several conventional water treatments, and it is resistant to chlorine, posing a serious threat to water sources. It can be eliminated through the filtering process. However, Cryptosporidium cannot due to their tiny size. Kindly don't forget to support my channel by subscribing to it. Characteristics of Giardia. It is a flagellated protist. It is diplomonad with tumbling mobility or two bodies with separate nuclei. Mitosomes take the place of mitochondria. It utilizes anaerobic glycolysis to metabolize. It has two stages, the reproductive trophozoite and the cyst stage mark the end of the life cycle. It is asexual reproduction. Giardia cysts are 9 to 12 mm long, and the reproductive trophozoites is 9 to 21 mm long. Giardia cysts are sensitive to chlorine, iodine, and chlorine dioxide as disinfectants. Its clinical manifestation of foodborne giardiasis. Giardia can be spread by 10 to 100 cysts, and symptoms generally appear 1 to 2 weeks after infection. The initial symptoms include gas, nausea, stomach bloating, abdominal cramps, and diarrhea. Dehydration and weight loss under extreme circumstances may result in persistent infection and possibly death. There are also situations when the patients are asymptomatic carriers but the illness only lasts a week or 5 days. Kindly subscribe to my channel. The life cycle of Giardia. Number 1. Giardia cysts can contaminate food, water, and surfaces, and they can cause giardiasis when swallowed in this infective stage of their life cycle. Infection occurs when a person swallows Giardia cyst from contaminated water, food, hands, surfaces, or objects. Number 2 shows you when Giardia cysts are swallowed, they pass through the mouth, esophagus, and stomach into the small intestine, where each cyst releases 2 trophozoites through a process called excitation. The Giardia trophozoites then feed off and absorb nutrients from the infected person. In number 3, you can see Giardia trophozoites multiply by splitting in two in a process called longitudinal binary fission, remaining in the small intestine where they can be free or attached to the inside lining of the small intestine. Number 4, the Giardia trophozoites then move towards the colon and transform back into cyst formed through a process called incitation. The Giardia cyst is the stage found most commonly in stool. And lastly, number 5. Both Giardia cysts and trophozoites can be found in the stool of someone who has giardiasis and may be observed microscopically to diagnose giardiasis. Giardia cysts are immediately infectious when passed in the stool or shortly afterward, and the cyst can survive several months in cold water and soil. Now let's understand the pathogenesis of Giardia species. Giardia's two-stage life cycle ends with the vegetative trophozoite, which multiplies and produces sickness, and the cyst that causes infection. The cyst outer filamentous layer, which is formed of N-acetyl galactosamine and 3 proteins called CWPs 1,3, which enable them to survive for a long time, makes them resistant to the temperature and environment, especially at low temperatures. When a cyst gets past the stomach acid and into the duodenum, it has 10 to 100% chances of infecting that area. Each cyst divides into 2 trophozoites upon entry into the duodenum, which populates the small intestine by consuming vital nutrients and allying the host. Giardia reproduces asexually. As a result, trophozoites are tetraploid of 4 sets of 1 chromosome and multiply through binary fission. When the cyst exits the stomach acidic environment and enters the duodenum and small intestine, excitation occurs and incitation then follows. The body's immune system activates these cells in reaction to the trophozoite, disrupting and shortening the epithelia microvilli. Additionally, it damages the epithelia tight junctions, increasing permeability and causing diarrhea. Additionally, the cyst undergoes bile salt and cholesterol deprivation before beginning to replicate, splitting in 2 trophozoites and infecting the other host. It is also clear that a strong immune response helps the host develop immunity to the illness. According to antigenic variation, the organism is said to have adapted to the various intestinal habitats. Some organisms with variant-specific proteins develop resistance to antibodies while not producing any poisons or virulence factors. Kindly show your love and support by subscribing to my channel. Epidemiology of Foodborne Giardiasis. The parasitic protozoan Giardia outbreaks affect 4 to 20 people per 100,000 people annually, and the majority are thought to be spread by fecal-oral contact. According to the Food and Agriculture Organization and WHO, out of 24 foodborne parasites, Giardia is listed in position 11 of these rankings. Giardiasis epidemiology is more common in developing nations and is connected to climatic and environmental factors. Children under the age of 5 are at greater risk than adults, and 80 to 60% of infections are asymptomatic. Chronic infections, however, account for less than 4% of cases. In Peru, 95 of the children under the age of 2 were infected, but 6 months after treatment, the virus returned. Children's early growth is stunted as a result of the condition, which is typically asymptomatic. Due to the excretion dose being larger than the infective dose each day, it was declared to be a waterborne outbreak. Although an infected individual can discharge up to 109 yeast each day, it takes 10 to 100 yeast to develop an infection. In impoverished nations where people drink tap water, shallow wells, and surface water, the potential for contamination is significant. The water sources are filled with zoonotic parasites that can infect humans when drank without filtration. Due to increased water intake and the fact that most affiliated children attend daycare facilities, the giardiasis infection often peaks in the late summer. A Minnesota outbreak was brought on by intake of home-canned salmon, which infected 60 staff members at a school. However, because asymptomatic giardiasis carriers were responsible for the outbreak, it was not classified as giardiasis. So what is the detection methods for Giardia species? Using a microscope to identify the Giardia cyst in stool samples is common practice, although it lacks specificity and sensitivity. Enzymatic immunoassays and immunochromatographic tests are two more amino acids techniques that are employed. However, they also lack specificity because of antibody cross-reactivity. The immunomagnetic technique of cyst separation is the most frequently utilized by the U.S. Environmental Protection Agency. The cysts are separated, then stained with fluorescent dye and differential interference construct to be seen under microscope. As it distinguishes the pathogens at the species and genotype levels, the PCR-based approach, when compared to other detection methods, offer increased specificity and sensitivity. So what can be the prevention and control measures of foodborne giardiasis? While asymptomatic infections are simple to cure, they are harder to find and must be monitored once they develop into chronic conditions. Nidazole, nitromedazole, metronidazole are the initial treatments of choice. In comparison to metronidazole, which takes 5 to 10 days to achieve the effective dose, nidazole is more effective since it has higher effectiveness in a single dose. Paromomycin is advised for pregnant women even though it is less effective than nitromedazole and non-absorbent aminoglycoside. Giardiasis in humans may be avoided by providing treated municipal water sources, which breaks the life cycle of parasite and stop it from spreading. Cysts and other pathogens are effectively eliminated by coagulation, clarifying, filtrating, and chlorinization processes used in municipal water systems. Giardia cysts can also be removed by using household portable filters with a size smaller than 1 meter, as well as heat treatments at 55 degrees Celsius and higher for 5 minutes. The risk of asymptomatic infection can also be reduced by food workers washing their hands and using protective gloves when preparing meals. Thank you so much for watching. Always keep learning, it keeps you young. Don't forget to like this video, subscribe to my channel and press the bell icon button so you don't miss any of my videos. Thank you so much.